Hello friends and my dear students. I want to give my introductions. Myself, Professor Mundhe Sandeep Janardhan, Assistant Professor and Head Department of Microbiology, Sanjeevanee Mahavidyalaya Chapoli, Taluka Chakur, District Latur. This college is affiliated under the Swami Ramanand Tirth Marathwada University, Nanded. As you know, due to the COVID-19 disease, it is not possible to conduct the classes in the college level, but due to the permission of UGC University and the government of Maharashtra, now the classes are conducting on online mode, and under the guidance of our respected principal Dr. Dhananjay Chati sir, our college is also conducting the classes on online mode. Today we will see the syllabus of BSc third year semester 5. This semester is based on choice based credit systems, that is the course structure. Before the study of syllabus, just we will check the marks pattern or how the marks are distributed paper wise. For semester 5 there are two theory papers. Paper number 12, Microbial Genetics, and paper number 13, Microbial Metabolism. These are the two papers under the semester 5. For microbial genetics, 10 marks for internal evaluations. Internal evaluations is the system where under college level the assistant professor they evaluated the internal marks, and 40 marks they are the end semester examinations. In this way 10 plus 40 is equal to 50 marks, or paper number 12, Microbial Genetics. Same pattern for microbial metabolism also, 10 plus 40 is equal to 50 marks. It means that 50 marks are for each paper. This is the sum marks criteria about the papers. Let us see the details syllabus of paper number 12 and paper number 13. Paper number 12 is the microbial genetics. Under the microbial genetics there are 4 units. We will see one by one the syllabus of microbial genetics. Unit first, it is the genetic material. Under this unit there are some points that we will see. The first point is the evidences for DNA as genetic material. Which evidences under the genetic materials? First, it is the Griffith experiment and Avery et al. Experiments, and second evidence or experiment is the Hershey and Chase experiment. The second point, discovery of RNA as a viral genetic material. Under that, the experiment, that is the Gerer and Scram experiment. This experiment is totally based on TMV, it means tobacco mosaic virus. Tobacco mosaic virus is the first virus forever discovered by the scientists Gerard and Scram, and they prove that RNA, it is the viral genetic material present in the tobacco mosaic virus. The next, properties of DNA as a genetic material. Next, chemical stability of DNA and its information content. Next, structure of prokaryotic chromosomes. Under that, Escherichia coli, the model genetic organisms. Unit 2nd, prokaryotic DNA replication. Under the unit 2nd, first point, general concepts of DNA replications. Second, semi-conservative DNA replication. C. Replication model, Keynes model. Then, precursors and enzymes of DNA replications. The next, mechanisms of DNA replications. Under that, initiation, elongation. In the elongation, beta clamp and progressive polymerase, and termination. And last point of unit 2nd, replication in Escherichia coli. In short, that we'll see about the Escherichia coli. Now we'll see the syllabus of unit 3, molecular recombination in bacteria. The first, general perspective of genetic recombinations with Holiday model as an example. Then, homologous recombinations of Escherichia coli. Under that, we will see initiation, synapsis, branch migrations, and resolution. Next point, types of recombinations, site-specific recombinations, it means interrogative and excessive recombinations. Second, illegitimate recombinations, it means non-homologous recombinations. And third, transpositions. Under that, first, transposable elements in prokaryotes, and B, insertion sequences. And last unit, for genetic exchange in bacteria. 1. Transformations. Under the transformations, we'll see the introduction and history. Second, mechanism of transformation. And third, competence, binding, penetration, synapsis, and interrogations. Second, conjugations. Under the conjugations, we'll see discovery of conjugation in bacteria. properties of a plasmid, that it means sex factors, HFR strains and their formations. Next, mechanism of conjugations, and last point, F factor and sex reactions. And under the unit 4, the last point, transductions. Under transduction we'll see subpoint, introduction and discovery, generalized and specialized transduction, and abortive transduction. So this is the syllabus of paper number 12, microbial genetics. So we'll see the syllabus of paper number 13, microbial metabolism. Under the microbial metabolism there are 4 units. Unit first, enzymes. Unit second, microbial metabolism. Unit third, mechanism of energy transformations in microorganisms. And unit fourth, microbial fermentations. Let us see one by one the syllabus unit wise. Unit first, enzymes. Under the unit first, enzymes, we will see subpoints. A, definitions, physiochemical properties of enzymes. B, coenzymes and cofactors. C, nomenclature and classification of enzymes. D, mechanisms of enzyme action. E, specificity of enzymes. F, enzyme kinetics, that is Micheli's-Menton equation. G, factors affecting enzyme activity. H, inhibitions of enzyme activity. There are different types of inhibitions: competitive, non-competitive and uncompetitive inhibitions. And last point of unit first, regulations of enzyme activity. Under that we will see allosteric enzymes, multi-enzyme systems and isoenzymes. Now see the second unit, microbial metabolisms. Under the microbial metabolisms there are different points we will see. The first, introduction to metabolisms, catabolisms and anabolisms with examples. B, role of nucleotides in metabolisms. Nucleotides as a building block of nucleic acid. ATP as a currency of cell, pyridines and flavin nucleotides. C, basic pathways of carbohydrates catabolisms, EMP pathway, HMP pathway, ED pathway and PKP pathway, TCA cycles. And last point, beta oxidations of saturated and unsaturated fatty acids. After that we will see the syllabus of unit 3rd, mechanisms of energy transformation in microorganisms. First point, respirations, photosynthesis and fermentations. Second, generation of ATP, under that oxidative phosphorylation, photophosphorylations, substrate level phosphorylations. Number C, biochemical mechanisms of respiration in heterotrophs and chemoautotrophs. D, respiratory electron transport chain in bacteria. And E, characteristics of bacterial, that is RETC and its component. Let us see the unit number 4, microbial fermentation. Under that, number 1, ethanol fermentation by yeast and bacteria. Number 2, lactic acid fermentations. There are 2 types of lactic acid fermentation, homo and heterolactic acid fermentations. Mixed acid fermentations, then acetone butanol fermentations. Next, butanediol fermentations, and last, succinic acid fermentations. This is all about the syllabus of paper number 12 and paper number 13. Okay, today this much part of the syllabus is sufficient. Tomorrow we'll see the details, informations about each point or each part of the syllabus. So friends, after the long gap, we are on the right track. It means we cannot stop education, or education cannot stop due to any problems. If it is not possible to conduct the lecture in the college level, no problems. There is sub options, that is online mode. We will conduct. We are conducting on onlines, and it's our duty to continue the educations. And one thing is very important, that you have to take, or everybody should take the precautions about COVID-19 disease, because it is a pandemic disease, it is microbiological disease, and COVID-19 disease is spreaded all over the world. It is uncontrollable manner, it is spreaded in the world. There is only one thing you have to do. You have to wear the mask, use sanitizer, keep social distancing between. It means that between the line you have to take the precautions, or everybody should take care about COVID-19 disease. We can't say how much time required for vaccine production, but we hope that in the recent times vaccine will available, and the problem of COVID-19 disease will solve. But it's our duty to live with COVID-19 disease by taking precautions. Thank you.